Hey guys, I want to do a quick little video going over parts of your DNA and replication digital interactive. So on page 1, we should be able to click on here and put our name in, please. Page 2 is just some directions. So when we're talking about DNA, we know that DNA is a long molecule that carries our genetic information. All right, so you can go through and use your notes that you should have already done to fill in some of this other information. On the next page, you're going to be talking about your different kind of bonds. Again, your notes should help you with this. Now, on slide 5, I want to help you out a little bit. Let's start with these little house-shaped figures. So with these little house-shaped figures, I think of the gingerbread house, and the gingerbread house is made out of sugar. So we're going to drag our deoxyribose sugar over there. And if this house is made out of sugar, this house is also going to be made out of sugar. Then we have, hanging off of that, the garage to your house, and that is where your nitrous vehicles live. So this is your nitrogen base, and your nitrogen bases come in a bunch of different varieties. So you can see we have 2 different varieties here. So they are A's, T's, C's, and G's in DNA. Then you have the little ball coming at your house, and then in lecture I talked about that being your little ball of poo coming to your house because the P in poo goes with the P in phosphate. So that is your phosphate group that is hanging off of your sugar. All right, and these make the backbone of your DNA, alternating sugars and phosphates. Here in the middle are your bonds. So these are your weak hydrogen bonds. All right, they're weak so that you can break apart your DNA in order to open it up and make copies. So we have 2 sides of one strand of DNA here, and you can see that this, with 1 phosphate, 1 sugar, and 1 base, this is 1 nucleotide. And over here on the opposite side, we have 1 sugar, 1 phosphate, 1 base. That is a second nucleotide. So let's move on to the next one. You are going to be comparing purines and pyrimidines, and you can also go back into your notes to look at what a purine and a pyrimidine is. And some of these things are going to be in the center, which is both. Like both of these have deoxyribose sugar. Okay, so go back in your notes where it compares these 2, and that will help you out with that. So for your DNA timeline, I'm going to help you with this. It all starts in the 1930s. With Oswald Avery, and he was the first guy who figured out that discovered that DNA was the molecule that carried genetic information. Okay, so before that, they didn't know if it was DNA or if it was proteins. They knew something was doing it. They just did not quite know what. All right. The next year, we have Chargaff. All right. Chargaff was in 1949. Oops, 49. And Chargaff is the guy who discovered, and we talked about this one in your notes, that there are equal numbers of A's to T's, because they go together, and C's to G's. If you remember from lecture, the apples go in the trees and cars go in the garage. For every garage, there must be a car, and for every tree, there must be an apple. Okay? All right, next we come to Rosalind Franklin. Rosalind Franklin was 1953. All right, and she was the lady who took X-ray photographs of DNA. And those photos were taken by her boss and brought to the next 2 guys, Watson and Crick, in the same year, 1953. And Watson and Crick were the 2 guys who discovered the structure of DNA, and they won the Nobel Prize for that. So, discovered the structure of DNA. Okay. All right, then a little later than that, and these ones we did not talk about in our notes, all right, but a little later than that, in 1977, we have these 2 gentlemen who they discovered how to sequence DNA, how to figure out where the A's, T's, C's, and G's are. So discovered how to sequence DNA bases. All right. So this was really important technology to get us where we are today, where we are starting to manipulate our DNA to cure genetic disorders. So in 2003, and you can see this is pretty recent science, this is where we first decoded all of the bases in the human genome. And so human genome is all of our genes. It's like the library of our genes. Okay, so it was in 2003 that that was accomplished. And here we are now in 2020 at the time that I record this. And we are already starting to manipulate this DNA and use it to do cool things. All right, on the next slide, we're going to be comparing prokaryote. Remember, euky, nuky, pro, no. So prokaryotes are bacteria, eukaryotes are animals and plants, and anything that's not a bacteria. So like in bacteria, for example, their DNA is circular shaped, whereas ours is linear. All right, so you're going to take the rest of these, and you can even use prior knowledge, or you can go ahead and Google if you don't know, and sort that information. On 4, you're just going to put the bases together. So remember, apples go in the tree. So these should bond together right there. And then cars go in the garage. So that one should go there. So you're going to put them together. Notice the number of bonds. There's 2 between A and T, 3 between C and G. So we're going to fill in the blanks on slide 5. Before a cell divides, it duplicates its DNA in a process called replication. We've been hearing that one over and over again. That happens in the S phase of interphase. During this process, the DNA molecule unzips into 2 strands that serve as templates. The enzyme DNA polymerase, there we go. Polymerase joins individual, this is going to be the monomers called nucleotides. Okay. Joins individual nucleotides to the original DNA strand following the rules of base pairing. Each new growing strand is called a complementary or matching strand. So we have complementary bases. All right, on page 6, we're going to just move these around as to what order they go in. So the very first thing that has to happen when you're looking at your DNA, we have a whole bunch of stuff, so you kind of have to move these around a little bit. So you have to be able to open your DNA first. I might have to, here we go. So the first thing is DNA helicase unzips your DNA, breaking the bonds between the 2 templates. So then you're going to have to decide which of these happens right after that. And of course you're going to end with 2 copies of DNA, right? So you're going to land up ending with this big one down here. So now you're going to take those middle ones and put them where they go. So use your notes, and they go through the steps of replication and finish that one. In the last 2, you're just going to make your complementary strands of DNA, similar to what we did before. So remember, apples in a tree, cars in the garage. And this one is the same thing. You're doing the other side of the DNA. All right? So when you finish that up, make sure you save your work and turn it in through Blackboard. Bye, guys.